Enzyme specificity. You have studied in your lower classes that enzymes are highly specific with respect to their action. This specificity is about the type of the substrates upon which they are acting, the type of the reactions they are catalyzing, the type of the product being produced, etc. In the following slides, we are going to talk about this specificity in detail. Specificity of enzymes. What do we mean by that term? It simply means that any enzyme cannot just act on any substrate or cannot carry out any type of chemical reaction. Enzymes can specifically act on some type of substrate or some groups of substrates and also can carry out only a particular type of chemical reaction. That's why we say enzymes are highly specific and selective. Broadly speaking, enzyme specificity is of two types, absolute and relative. Some type of enzymes can act only on a particular substrate and we call it absolute specificity. For example, glucokinase can act only upon glucose. Glucose reacts with ATP forming ADP and glucose 6-phosphate. Another example is that of urease. So these enzymes can act only upon that particular substrate molecules and no other molecule. While coming to relative specificity, here the enzyme can act on a given class of substrates. And that class of substrates includes many substrates. For example, hexokinase can add ATP to glucose, fructose, mannose, etc., which are 6-carbon sugars. So it is less specific, we may say, compared with the glucokinase. Another example, peptidases. Actually, remember, there is no enzyme called peptidase. Peptidases is a class of enzymes. Peptidase can act on peptide bonds in different proteins. Remember, hexokinase is the name of a given enzyme, while peptidase is not a given enzyme, it is a class of enzymes. Some enzymes are specific not only to the type of the bond they are producing, but also to the chemical structure surrounding that bond. I mean the bond they are producing or breaking down. Good example is pepsin. Pepsin is an endopeptidase which is breaking long protein molecules into smaller fragments. The importance of pepsin is that it will act on any chemical bond where the amino group is contributed by aromatic amino acids only. For example, phenylalanine, tyrosine, tryptophan, etc. are aromatic in nature. And when these amino acids are contributing their amino group to the peptide bond, then pepsin can digest that bond. Remember, pepsin is a hydrolase. While if the amino group is being contributed by serine or other amino acids which are not aromatic in nature, then pepsin will not act upon it. Group specificity. Optical specificity, also called stereospecificity, is a particular type of specificity in which the enzyme can accept only one of the stereoisomer or one of the optical isomer, either L or D. What is given in this slide is about lactate. This lactate dehydrogenase can accept only the L form but not the D form. And in the next slides you can see the other examples as well.
Optical or stereo specificity. You can see here, this is the enzyme. This enzyme is called L-alanine oxidase. This is L-alanine oxidase, and this is L-alanine. When L-alanine is given to the enzyme, there is enzyme action. Look here, in this diagram, D-alanine is supplied to the enzyme and there is no action. So this is a classical example of optical specificity. You know that D and L forms are different isomers.
We have amylase enzyme in our body. In our saliva we have that enzyme, and in the pancreatic juice also we have amylase enzyme. Our diet contains starch. The starch is made up of alpha 1,4 glycosidic linkage, and the starch can be digested by the amylase. But whenever we are taking vegetarian food material, vegetable matter, the cell wall of the plants is made up of cellulose. Cellulose consists of beta 1,4 glycosidic linkages, and this cellulosic material is not digested in our body. Why? Because alpha amylase shows optical or stereo specificity. It can digest starch while it cannot digest cellulose. Ruminant cattle are able to digest cellulose because in their rumen some microbes are there. These microbes are producing cellulase enzymes, which is specific for beta 1,4 glycosidic linkage.
Stereo or optical specificity. Geometric specificity. The enzyme can act on different substrates with similar molecular geometry. For example, let us take the case of ADH, alcohol dehydrogenase. ADH, alcohol dehydrogenase. It is acting upon alcohol and convert it into the corresponding aldehyde. Look at the diagram. You can see ethanol is converted into acetaldehyde while methanol is converted into formaldehyde. You can see ethylene glycol as well, which is converted into glycolaldehyde. Here, the same enzyme can act upon a given class of substrates. So this is a very good example for geometric specificity. And you know that this aldehyde formed is responsible for the various physiological actions once you consume alcohol. You know that the whiskey, brandy, rum, etc. contains ethanol as the alcohol, while methanol is often an adulterant and it is highly toxic also. It is actually added during adulteration into this alcohol, which is not at all recommendable.
Coenzyme specificity. You know that coenzymes are non-proteinaceous molecules required for the optimum functioning of many of the enzyme molecules. Remember, just any coenzyme cannot interact with any enzyme. They are highly specific in this case. Let us take an example. Both FAD and NAD are hydrogen carriers, and they are acting as coenzymes for different types of dehydrogenases. Let us take the case of succinate dehydrogenase. We have studied this enzyme in Krebs cycle. Succinate dehydrogenase is using only FAD and not NAD as the coenzyme. FAD is receiving hydrogen from succinate, thereby forming FADH2, and the succinate is oxidized to fumarate. Succinate dehydrogenase is highly specific in that it will accept only FAD as the coenzyme. Glyceraldehyde 3-phosphate dehydrogenase is an enzyme in glycolysis. It acts in the only stage in which NADH plus H plus is produced during glycolysis. This enzyme can accept only NAD and not FAD as its coenzyme. This is another case of coenzyme specificity.